Prudent Scholars in association with Goa Headmasters Association.
Hello everyone, last time we have covered autotrophic nutrition. In today's session, we are going to cover heterotrophic nutrition. We already know what are heterotrophs. Heterotrophs are the organisms which are dependent on other organisms for food. Heterotrophs, they will not prepare their own food. And the mode of nutrition in heterotrophs we call heterotrophic nutrition. Heterotrophic nutrition is a type of nutrition in which organisms depend upon other organisms for food to survive. These are some of the examples. One of the examples is fungi. Heterotrophic nutrition takes place in animals, it takes place in fungi. First, we will consider fungus. These are the two examples of fungi, bread mold and the other one is yeast. One more example of fungus is mushroom. Now, fungus, what they do? They normally take food from the dead and decaying matter. So, fungus, they will secrete enzyme and that enzyme will break down the complex substances outside their body. And once those complex substances are broken outside their body, they take in simple substances. For our understanding, one of the examples is of complex substance is glucose for our understanding and simple substance is sugar. So, fungus, they break down complex substances outside their body using the enzymes. Enzymes acts on those complex substances and they break down complex substances and simple substances are taken in by the fungus. This mode of nutrition in fungus, we call it as saprophytic nutrition. Saprophytic nutrition comes under heterotrophic nutrition. In this way, fungus, they absorb the simple compounds. Now, there is one more type of heterotrophic nutrition, we call it as parasitic nutrition. There are some organisms which are known as the parasites. And these organisms take food from the other organisms without killing them. Such examples are orchids, Bile, also known as the cuscuta, then ticks, which are found on dogs and all, orchid which grows on other plant. There are many orchids and they bear beautiful flowers. Bile, which is found on other plants. So, lice which are found on human hairs, leeches, they suck blood, then tapeworm, which is sometimes these tapeworms, they are found in human intestine. So, these are the examples of parasites and the mode of nutrition in such organism is parasitic nutrition. Let us understand nutrition in amoeba and nutrition in paramecium. Both are unicellular organisms. Amoeba, paramecium. Let us focus on amoeba first. So, amoeba, we have seen amoeba. Amoeba does not have regular shape, it has irregular shape and we call these extensions of amoeba as pseudopodia as we can see here. So, amoeba has nucleus, this part of the amoeba we call it as cytoplasm. Amoeba, when amoeba detects food particle, amoeba will try to move towards the food particle and using these pseudopodia, amoeba will try to cover that food particle. Amoeba will take in food particle. When amoeba takes in that food particle, temporary stomach will be formed, and temporary stomach in amoeba is known as the food vacuole. As we can see here, food vacuole. This food particle, food vacuole is formed. In this food vacuole, enzymes are secreted. So, enzymes enter in this food vacuole and they act on this food particle. We have learned regarding enzymes in 9th standard. They are the biocatalyst that helps to break down the substances before substances are taken in by the body, or before these substances are utilized by the body. So, enzymes acts on these food particles. These enzymes will break down the food particle. Digested food will be absorbed by the amoeba. Digested food will enter in this cytoplasm. Undigested food will be thrown out by the amoeba. So, in this way the amoeba takes in food, but paramecium will not take in food from any part. Amoeba can take in food from this part, from this side, any side, because the entire surface is in contact with the environment. Although paramecium is unicellular, I repeat, although paramecium is unicellular, paramecium has halo-like structures called as the cilia, which helps in making movement. And paramecium takes in food at specific spot, not at any place, not at any spot. And using cilia, paramecium will try to bring the required food material here, and we can see here cell mouth. So, in case of paramecium, food is taken in at specific spot. Let us understand the difference between amoeba and paramecium. Amoeba has irregular shape, paramecium has definite shape. Then food is taken in at any spot and food is taken in at specific spot in case of paramecium. So, this is regarding the amoeba and paramecium. Now, we will learn nutrition in human beings. So, there is a long tube which starts from mouth and ends at anus. We call this long tube as alimentary canal. So, we can see it starts from here, alimentary canal, and it ends at anus. This diagram can be asked in the examination. Nutrition in human beings. When we take in food, at that time in our buccal cavity, in our mouth, there are teeth. So, teeth, they crushes the food, they break down the food physically. Then in our buccal cavity, as soon as we take in food, saliva will be produced, and this saliva is produced by gland known as the salivary gland. So, this salivary gland produces saliva, and this saliva consists of enzyme. There are many examples of enzymes in human beings. For example, the enzyme present in saliva is known as the salivary amylase. This salivary amylase plays very important role. It breaks down starch, which is complex substance in our mouth, which if it is present, this breaks down starch into sugar. So, salivary amylase breaks down starch into sugar. Sugar is a simple substance, starch is an example of complex substance. Glucose, starch, they are known as carbohydrates. So, breakdown of starch takes place in our mouth, breakdown of starch takes place in buccal cavity. It takes place here. As we can see here, tongue also plays very important role. Tongue helps in mixing the saliva thoroughly with the food. So, tongue has its own function, then teeth crushes the food, salivary gland secretes saliva, then food will enter from mouth to stomach and the food will pass from mouth to stomach through esophagus. So, esophagus transfer food from mouth to stomach. Esophagus is also known as food pipe. Now, the movement of food takes place right from this point up to here. Okay. This movement is known by special name. What happens is, let us consider this esophagus. Muscles are present here. So, these muscles which are present here, okay, these muscles will contract rhythmically and because of this contraction and also they will relax. Because of this contraction and relaxation, the food will pass and we call such movement as peristaltic movement. We call such movement as peristaltic movement. Now, food enters in the stomach. In stomach, the walls of the stomach consist of glands known as the gastric glands. So, these glands, gastric gland, secrete juice and gastric glands secrete juice such as hydrochloric acid HCl, then mucus and pepsin. Each has its own function. Hydrochloric acid, when food enters in the stomach, hydrochloric acid kills the germs that enters in the stomach along with the food. So, hydrochloric acid plays very important role. It kills the germs that enters in the stomach along with the food. Hydrochloric acid also makes this medium acidic. In short, hydrochloric acid kills germs in stomach and it also makes the medium acidic in stomach. Now, once this medium is made acidic, this helps enzyme, pepsin is the name of the enzyme. This enzyme digests proteins in stomach and making medium acidic facilitates the action of enzyme pepsin to digest proteins. So, pepsin digests proteins in stomach. Then we have mucus, protects the inner lining of the stomach from the action of acid. So, acid may harm the inner lining of the stomach. Under normal conditions, mucus protects the inner lining of the stomach from the action of acid. So, these are the things, these are the functions of hydrochloric acid Mucus, as I said just now, it protects the inner lining of the stomach from the action of acid under normal conditions. Pepsin, this is the name of the enzyme which digests proteins. So we have seen, we came across two enzymes now. One enzyme is salivary amylase, which breaks down starch into sugar, and one more is pepsin, which digests proteins in stomach. Next part. So from stomach, food will enter in small intestine. Small intestine. Now, from stomach, food enters in the small intestine in small amounts, and this is regulated, this is taken care by the muscles which are present where stomach and small intestine meet. We call such muscles as sphincter muscles. So from stomach, the entry of food in the small intestine, this is regulated by sphincter muscles, as we know. Now, small intestine of herbivores and of carnivores is of different length. For example, we already know the herbivores are the animals which eat plant or plant products. Carnivores are the animals which eat meat. One of the examples of herbivores is cow. So their small intestine is longer, and of carnivores, small intestine is short. The herbivores, they normally eat grass, and grass consists of cellulose. Cellulose is not easy to digest. So to allow cellulose to digest, herbivores have longer small intestine, and carnivores, they eat meat, and meat is easy to digest, and that is why carnivores have shorter small intestine. Once the food enters in the small intestine, when they enter, the food was acidic. Okay. But in the small intestine gets the secretion from liver. Small intestine also gets the secretion from pancreas. I repeat, small intestine gets the secretion from liver, small intestine also gets the secretion from pancreas. So liver, as we already know, liver secretes bile juice. Liver secretes bile juice. Bile juice makes the medium in small intestine alkaline. So bile juice makes the medium alkaline, and this will help the enzymes which are secreted by pancreas to act properly. Pancreas secretes pancreatic juice, which consists of two enzymes known as trypsin and lipase. Once the medium is made alkaline, I said just now that bile juice makes the medium alkaline, and once this medium is made alkaline, those enzymes can act. The enzymes present in pancreatic juice which is secreted by pancreas, those enzymes can act properly. So bile juice, let us understand the function of bile juice. So bile juice makes the medium alkaline. Besides this, in small intestine, large globules of fats are present. So, if large globules of fats are present, bile salts present in bile juice, they can act as we can see here. So, large fat globule, bile salts act on these large globules of fats and they break down these large globules of fats into small globules. And this process of conversion of large globule of fats into smaller globules of fats by bile salts present in bile juice, we call this process as emulsification of fat or emulsification of fats. Coming back to this diagram, regarding the small intestine, we have already covered the functions of the bile juice. Okay. Next, pancreas secretes pancreatic juice, and pancreatic juice consists of two enzymes, trypsin and lipase. Trypsin digests proteins and this happens in small intestine. We have seen earlier two enzymes, salivary amylase and pepsin. Pepsin digests proteins in stomach, trypsin digests proteins in small intestine. Lipase, what lipase does? It also performs function. So, lipase can act, they break down the small globules of fats. So, function of the lipase is lipase breaks down the emulsified fats. So, we have covered. So, we have seen just now regarding the lipase and trypsin. Trypsin digests proteins in small intestine. Lipase digests emulsified fats in small intestine. So, in small intestine there are walls and in small intestine there are intestinal glands, and these intestinal glands they secrete intestinal juice. So, these intestinal juice consists of enzymes which further breaks down carbohydrates into glucose, then fats into fatty acids and glycerol, proteins gets broken down into amino acids. So, complete digestion takes place in small intestine. If there are undigested food, so that undigested food will enter in the large intestine. But small intestine, as I said earlier, it is the site or it is the area where it is the place where the complete digestion of food takes place. Now, small intestine consists of finger-like projections, and we call finger-like projections as villi, as we can see here. So, the walls of the small intestine consist of finger-like projections called as the villi. This provides the surface area for absorption of the digested food. Just now we have seen that complete digestion takes place in small intestine. The digested food is absorbed in the small intestine and villi provides the surface area for absorption. So, these villi, they are richly supplied with blood vessels, and those blood vessels, they will carry the digested food to the required part or required place. So, that digested food can be used to produce energy, then to repair new tissue, to build new tissue. From small intestine, undigested food enters in the large intestine, as we can see this large intestine, and in large intestine again villi are present. They will also absorb the water from this undigested food, okay, and undigested food is given out through this part called as the anus, and there are muscles called as the anal sphincter muscles. One more thing regarding the stomach. There are walls present in the stomach, and these walls helps in mixing the food thoroughly with the gastric juice. This is the function of walls, muscular walls present in the stomach. So, in this session what we have covered? We have covered heterotrophic nutrition, we have covered nutrition in fungi, nutrition in amoeba, we have covered nutrition in paramecium, we have seen the difference. After this, we have completed nutrition in human beings. One more thing to tell you, that I told regarding the peristaltic movement. So, peristaltic movement, it occurs right from this food pipe till anus, okay. So, many questions can be asked based on this topic. In fact, any question can be asked from our science textbook. So, this statement is very important for us. So, at home we have to read and understand the lessons, we have to read lessons many times. So, these are the things that we have covered today regarding the parasitic nutrition also. Diagram of nutrition in amoeba can be asked in the examination, how amoeba takes in food, difference between amoeba, paramecium, okay. Then we have seen the function of teeth, what salivary gland secretes, which enzyme is present in saliva, function of salivary amylase, function of saliva also. Saliva makes the food wet. Function of tongue, diagram of human alimentary canal, as I said before, can be asked in the examination, okay. Yes, about gastric glands. So, these are the things that we have covered in this session. Carnivores have shorter small intestine, herbivores have longer small intestine. So, these are the things that we have covered. Thank you.